Dr. Dow, welcome back to the Tick Bootcamp podcast.
Thanks, Rich. It's great to see you again, and nice to meet you, Matt.
We are excited to have you. And I did share with Matt when I had the speed dating interview with you over at the ILADS conference last year that we needed to have you on for a long form podcast. And as it turns out, it took almost a whole year for us to get you back, but I'm happy we were able to do that before we saw each other again at the ILADS conference.
Well, it sounds like a good first date to me.
Yes, well, it was. The speed dating was rough. It just as an old married man, having 61 speed dates over 2 days was a very interesting experience at ILADS, and
No.
I look forward to repeating that experience again for the folks in the community. It was the ILADS series that we did was very well received, and your last episode was very well received, and that was by the way, for folks who want to get a little more background on you, that was episode 463 from our ILADS series, where we first met Dr. Dow and had a very nice introductory interview. But we're going to go into some more detail now, Dr. Dow. So why don't we talk first about your background? Folks have probably discerned that you have a slight accent. It's not a heavy accent, but a slight accent. So where are you from?
I'm from Perth in Western Australia originally. Did all my grad training there, and then I moved to Washington, D.C. in 2000. So I've been here like 25 years or so.
So let's talk about your grad training. What did you study as an undergrad, and what did you study to earn your PhD?
So my undergrad was in biotechnology, and then I did a PhD in malaria drug discovery and development.
All right. And of course, folks in this community are probably anticipating what it is we're going to be talking with you about since you did your uh, graduate research in the malaria arena. But let's bookmark that for a second. Talk a little bit about where you're currently working and what's the name of the business you developed.
Yeah, so I was a Army contractor at Walter Reed Army Institute of Research for about 10 years. And then when I left the rare in the early 2010s, I founded 60 Degrees Pharmaceuticals. I remain the CEO and a board member, and we're in the process of a pretty interesting clinical trial program in Babesiosis with our lead product, which is Arakoda, which is FDA-approved for malaria prevention. And then we're taking a look at the active molecule in that product, Tafenoquine, for babesiosis.
Okay, so again, let's pause there for a second, and I want to build out the window of time when you had done the research at the U.S. Army's anti-malarial drug treatment program. So talk to us about that. How long a period of time were you doing that work, and share with our listeners what insights you learned as a postgraduate.
So at Walter Reed, I started out as a postdoc, and then I kind of um, grew into a role of increasing responsibility, leading up to being a senior technical advisor in the clinical development program at Fort Detrick. I worked on lots of different molecules to treat and prevent malaria while I was there. So I'd say I got a fairly good grounding in, basic concepts in drug discovery and development. And because of the nature of the organization, I wasn't pigeonholed doing one thing, and which is not the case if I'd taken a career in industry where you tend to be focused on one specific element of the drug development process.
So, and if it wasn't enough that you already had, your bachelor's degree and your PhD, you also earned another graduate degree during that window of time here in the U.S. So, talk to us about your additional graduate studies here in the U.S.
Yeah, so I'll confess I was a total science nerd, and many people would say I still am. And I think you do a PhD in a very specialized area, and you are sort of trained to feel like um, you're the center of knowledge and expertise in that particular area. And I was concerned about the ability to have credibility and be able to communicate with folks with a more general business background as I was contemplating starting a company and moving into a different field. So I found an MBA beyond the technical skills you learn, just very socializing as I was transitioning from kind of very data and science focused to necessarily having a more general perspective on things when you move into running a business.
So you certainly have many titles. In fact, you have a very short last name, DOW, and then it's BSc,
Yeah.
MBA, PhD. So you have a whole bunch of letters after your name that are much longer than your name itself. There, it's an accomplishment when you have more letters after the comma than you have before.
Well, I don't think I think it's more about what you accomplish after putting in all that hard work than the credentials themselves. And I hope as we move from malaria prevention, which is our core business, increasingly more into tick-borne disease, that we'll have the opportunity to help a lot of patients with new therapies. So that's what I'm focused on. Um, it's just a fact that you need all the training in addition to all the life experience to really be successful in a niche field. And so I hope in the next few years I will be fortunate enough to really impact some folks' lives as we roll out these new therapies.
All right. Well, I appreciate the humility, but I'll make believe I'm your dad, and I'll brag for you because of all of the educational accomplishments you have, and I appreciate you dedicating not just your education, but your focus on tick-borne diseases and trying to help folks who are suffering from these diseases. I mean, we've interviewed hundreds of patients,
Yeah.
and folks know. We're very passionate about this disease because it does really disrupt lives. So thank you for dedicating your focus to this underserved population of folks. And so now I'd like to, and Matt's going to join us now. I'd like to move into the parallels here between malaria and Babesia.
Okay.
And one of the things that Matt and I have observed over the 5 years we've been doing this podcast is, um, very early on, both we and the community were very Borrelia focused, and
Yeah.
most of the people we interviewed early on who were suffering from chronic Lyme disease were really, at least from a diagnostic standpoint, from a testing standpoint, were largely diagnosed with a single bug, a single bacteria. And what we're seeing
Yeah.
now is that's very rare. Now we're seeing people with a polymicrobial infection, which is why we've changed our definition of Lyme disease here. And we almost always see the three Bs: Borrelia, Bartonella, and Babesia. And we're really excited to have you today because we really don't have a lot of folks focusing on Babesia, and that's something we've wanted to build out, which is the main reason we're excited to have you here on the long form podcast rather than the short form we did together at ILAD. So, can you first help us to gain some insight into why we are making the observation? I'm wondering if you're making the observation that there seems to be a substantially greater number of people who are diagnosed with an illness that includes Babesia.
So I might just take a step back a bit and share where I came from with malaria and what I've learned about Babesia as I've kind of entered the space, and then I'll get to your question in a long way. So
Okay.
in some respects, malaria as a disease is really straightforward, right? It infects red cells. your red cells burst, you get a flu-like illness, you get anemia because your red cells are getting chewed up, and then that can lead to severe disease. But from a treatment standpoint, there's been 100 years of research in anti-malarial drugs, and we've got it down to 3 days of dosing, and that clears up almost everybody's infections unless there's a drug-resistant strain. And so in the malaria business, it's an exercise in the constant war against drug resistance and replacing parts of the old combinations with new agents. So, that naturally leads to the idea that anything that works for malaria works for babesiosis. And I think that's what drove some of the Babesia physicians to try Tafenoquine out as an addition to standard of care in immunosuppressed patients, and led to a whole bunch of non-clinical research from some of the academic folks that paved the way for that. But the fundamental difference, and there's a lot of commonalities in the kind of disease cycle with red cell turnover, flu-like illness in the acute phase of the disease, and common drug targets, which means the extrapolation from malaria drug to Babesia drug kind of makes sense. But where the difference comes out is in really the chronic disease and the mystery around that part of the illness, and that from a scientific perspective, and then maybe try and relate it back to the patient experience. So if you've got acute babesiosis and you're immunocompetent, so nothing wrong with you, otherwise healthy, you're less than 50, you've got a spleen, you're not taking any immunosuppressive drugs, the dogma is the standard of care works, you recover from your clinical symptoms, and whatever parasites might be left aren't really a medical issue. And
so, Doctor, can I ask you to just explain that in more detail? When you say the standard of care, does that mean that the person who is infected with Babesia, with all things being equal, if they have a healthy immune system, that they'll be able to clear the parasites without any medical intervention, or will you need medical intervention in order to be able to clear the parasites?
Yeah. So in folks who've never had babesiosis before and have a normal immune system, completely normal, and you get symptomatic with the disease and your infection has been confirmed with microscopy or a PCR, this is standard Infectious Disease Society of America stuff, and we'll get to that in a second. That professional society recommends atovaquone and azithromycin, so Mepron and Zithromax, as the combination agent to treat symptomatic babesiosis. And the dogma, which I think is mostly an assumption because there isn't a whole lot of literature on it, is that in folks with a normal immune system who are treated with that standard of care regimen, the assumption is that it either kills all the bugs, or if there are any bugs left, they're not that clinically important. So that's kind of the dogma. Then there's a group of folks who are immunosuppressed or at risk, and they're folks who don't have a spleen. They might be taking immunosuppressive drugs for a transplant. They might be on a B-cell depleting antibody because they're being treated for an autoimmune disease, or they might have cancer. That's sort of that's sort of the cluster of risks for people who can experience severe disease or persistent disease that is recognized and known to persist for months or years in that specific group of patients.
So, can I
And
quick question on that,
sure.
Dr. Dow? Because, I understand that you're saying people that are immune compromised or the elderly, they are more susceptible to a chronic infection or a relapsing infection of Babesia. But I can't help but wonder that so many of us in the Lyme community are bit by a tick and we get multiple infections at once. So would a
Yeah, I'm
healthy
getting to that.
person, okay, I'm sorry, I'm getting ahead of
I'm
it. I'll let you keep going.
I'm getting to that. So
I'm messing with your groove.
yeah, no problem. And part of framing it this way is because I'm trying to frame it in a way that that gives you a scientific insight into the way conventional folks think about the disease and then where the gaps might be in the knowledge, right? So high risk factors, severe disease, persistent disease, this is well documented in the literature and not controversial. Where there's been literally zero papers is looking at folks who have dysregulated immune systems, right? So not folks who have, quote, normal immune systems, but have been dysregulated for various reasons. So in that bucket of folks, you can include people with long COVID, people with chronic fatigue syndrome, people with post-treatment Lyme disease, and all of which have overlapping symptoms, as you know, but also have the common theme amongst them that the immune system is not the same as in a person that's young and healthy and doesn't have any of those prior medical experiences. And so the hypothesis that we're testing in our clinical trial program with the chronic disease study that will start at the end of October is that essentially there's a group of folks with dysregulated immune systems who are more vulnerable to clinical or disease symptoms from a persistent Babesia infection. And even more specifically, it compromises your ability to recover from the symptoms of chronic illness caused by something else. So that gets back to your original question, which is tick bites and multiple disease pathogens contributing to people's symptoms.
So, Dr. Dow, can we walk back a little bit more? Because I just want to be clear. So, first of all, the parallels between malaria and Babesia. What is the reservoir for malaria versus what is the reservoir for Babesia? And then what is the vector that brings the malaria from the reservoir to the human
Okay, yeah.
population versus the vector that will bring, or vectors that can bring the Babesia from the reservoir to the human population.
Yeah, so I was focused on what happens when the parasites are in your system already. Of course, malaria is transmitted by mosquitoes, and a very specific kind of mosquito called an Anopheles mosquito. And geographically, the mosquitoes are present anywhere it's warm and humid. And, in the US specifically, the natural range seasonally is right up to the Canadian border.
Okay.
And the historically actually used to be a lot of malaria in the southern United States, up until the first half of the 20th century, where it was eradicated with pesticides and fly screens and public health measures. So
So
it's
what?
mainly now a risk to travelers, and then occasionally you get someone come back from a place with malaria, and then they'll get bitten by a local mosquito that will transmit the infection to other people, just like it used to 100 years ago. And that's why you hear occasionally people in Florida or Alabama, or I think there was a case in New Jersey recently, where there's malaria transmitted locally in the United States.
So, Doc, now what's the reservoir for malaria? Meaning, where does the bug come from prior to the mosquito biting the human?
A human reservoir. So following an infection you the parasite forms sexual stages called gametocytes, which can persist for weeks or months. And that's when the mosquito bites you. And those particular replicating forms of malaria don't cause any symptoms as far as we know. So when the mosquito bites you, it ingests those sexual stages, which then form together and create new malaria parasites. And then those are transmitted to the next person the mosquito bites.
Now, can malaria be transmitted from human to human without the mosquito vector?
Um, I suppose it could be transmitted through the blood donation system,
Okay.
but that would be incredibly rare in the United States, where there's almost no existing parasite reservoir at the moment.
So it cannot be transmitted sexually, and it
It
cannot
cannot,
be,
no.
and it cannot be transmitted mother to child.
I think that the main risk with pregnant women is to the fetus from the infection itself. And I don't actually know what the risk is of, transmission to a fetus itself from a pregnant woman. It's usually the thing we worry about is the loss of the fetus as a consequence of the malaria infection itself. Yeah.
So you don't know if it can be congenitally transmitted. What
I don't know
about
the answer to that. No.
what about breastfeeding mothers? Can a breastfeeding mother not through breastfeeding? Okay.
No.
So now let's focus on Babesia. How is Babesia? What is the reservoir for Babesia? Meaning, where is it in the either human or animal population? And what are the vectors that can transmit Babesia from whatever the reservoir is or reservoirs are to the human population?
So I'm not an entomologist or a tichologist or whatever the professional title for those folks is. So I'm just going to simplify it to my working knowledge of it, which is essentially Babesia is transmitted by ticks, so specifically deer ticks. The dogma is it's mostly in the Northeast and the Midwest, although when you look at medical claims data for babesiosis, it's a lot broader than that in terms of the geographic localities, and it is a zoonotic infection, meaning that there's a cycle in mice and deer and other animals as well that the ticks are. Mosquitoes with malaria, they tend to be very specific to humans. There isn't a whole lot of mosquitoes that bite other animals and then bite humans. But with ticks, that's not true. They tend to be a bit less selective, and that's why you get this transfer over from deer and mice into humans.
Okay. So you are now building out. Before I asked you to walk back and help us with the reservoir host and vector cycle, you were beginning to build out the parallels between malaria and Babesia, and then the distinctions between the two. So help us with the parallels first, and then build out the distinctions for us, so that we can get a sense of why you're doing the work that you're doing now, and why the clinical trials and the research relating to malaria is not necessarily going to be instructive in treating Babesia.
So the most obvious thing is that they're both parasites of red blood cells, right? And it's the red cell destruction when they multiply inside the red cell and then burst out that causes all the acute symptoms of both diseases. Of course, what transmit them, we've just talked about, is different. And so there's a lot of common biology when you're a red cell parasite but differences, which is why we see some of the drugs used as antimalarials have some activity against Babesia, but they're not automatically wonder drugs just because they worked against malaria. So, a 3-dose regimen of Coartem, which is an FDA-approved drug for malaria, is not going to eradicate a Babesia infection. So with our drug specifically, the rationale for why it was used is because for the class of drugs it comes from is called an 8-aminoquinoline, and it's different from, say, chloroquine or hydroxychloroquine, which everyone's heard about, because of the placement of some of the side chains on the core ring. And that may sound like scientific blah blah blah, but it's important because it really confers a different mechanism of action. So in the case of 8-aminoquinolines, they induce oxidative stress inside red blood cells. And they seem to do that with Babesia as well. So that was one of the arguments for trying it out, both because it's similar between malaria and Babesia, so in theory it should work, but also because it's different from the way the existing drugs used for treating Babesia work. So azithromycin works through an inhibition of protein synthesis, and then Mepron works by disrupting the electron transport chain, whereas so just different biochemically, and that's important for thinking about drug resistance in the context of both of the standard of care agents, which are highly susceptible to resistance depending on how they're used.
I have another off-topic question that I wanted to ask you, and then we can return to this, and that is, can you share with me and our listeners what is sickle cell anemia? What is its relationship to malaria? And is there any connection between immunity from Babesia and sickle cell?
That is a fascinating question. So there are 2 enzyme deficiencies. One of them is sickle cell anemia. No, let me back up. There are 2 genetic conditions that cause one, sickle cell anemia, and then separately G6PD deficiency. And those 2 genetic conditions evolve because they confer some resistance to malaria, which is why you see a lot of sickle cell anemia in Africa, and then a lot of G6PD deficiency historically where there's been a lot of malaria.
And we see it with a lot of folks in the Black community here in the U.S.
That's correct, yeah. So what we don't know, because I don't know that anyone's ever looked, and that might reflect my lack of knowledge about specific literature, which I've not had a professional reason to go and do a deep dive into. But my suspicion is there's not much known about whether those 2 enzyme deficiencies would correlate with more or less susceptibility to babesiosis. Although the hypothesis that you take from the analogy to malaria is, a reasonable one, I think, to go and study if you were interested in that.
All right, so now thank you for indulging me there, because it was the burning question I had in my head before we started the interview today. So let's come back now to the work that you're doing and the clinical study that's starting in October. And I think there's also the possibility of a couple of other clinical trials in the very near future. So frame for us the clinical trial that you're beginning in October, and give folks who are listening to this interview some sense of what your goals are with regard to treating people who have a polymicrobial infection, and contrast that from people who are immunocompromised for some other reason and therefore unable to manage the parasites in a way that they would be able to if they were not immunocompromised from something other than a tick disease.
Yeah. So this is a real can of worms, as I'm sure you can imagine. So let me just start with very brief descriptions of the 3 trials that we're doing. So Arakoda, which is our FDA-approved molecule for malaria prevention, is commercially available in the US, and it has been the subject of 20 plus clinical trials over 30 years and has a pretty well established safety profile. And for that reason, when we go and talk to the FDA about a new labeling for our prescribing information for babesiosis, our assumption and hope is that the concerns about safety, which you usually have for a new disease, are perhaps going to be less than they would be if we were, for example, going to a higher dose, which hasn't been as extensively studied. So the first thing to say is that we're investigating the dose with an established safety profile in babesiosis, and we're very intentionally not increasing the dose because of that safety profile.
So this is purely just evaluating
Off-label.
the off-label use.
It's it's obvious from our sales data, from talking to people in the community, that Tafenoquine is being used off-label in babesiosis. But one of the challenges for the company is it's being used in all kinds of ways at all kinds of different doses. And so part of our clinical trial program is to prove out what we believe, and we hope the FDA will agree with this, is a safe dose for babesiosis, but to do it using a series of clinical trials that show that. And then we've stuck to the dose that's already approved for malaria because it's less risky, both for patients, and it's less risky for the company because we don't have as many arguments to have with the regulator about moving forward.
Dr. Dow, just a quick question for you, because is your use of Tafenoquine just for severe persistent infections, or would you recommend this potentially for acute infections as well to ensure it's eradicated even in a quote unquote healthy patient who maybe won't respond as well to standard treatment as one would think?
So, of course, I can't recommend anything.
Oh, yes.
Yeah.
Yeah.
I have to be very clear about that. And I also have to say, and this is to protect me and the company legally, that Tafenoquine and Arakoda are not approved by the FDA for babesiosis. And what I'm about to outline is the series of trials that we want to do so that we can go and have that discussion with the agency about it.
Got it.
We have 3 trials focused on severe disease. That's one, that's a randomized study. We have an open label expanded access study in patients who are immunosuppressed, classically immunosuppressed and failed prior therapy. And then we have a 3rd trial, open label trial in chronic disease, which I'll describe in a minute. Those are the 3 populations we think where the highest unmet medical need is.
Yep.
And where the argument to practitioners and regulators should be the most straightforward. How the drug ends up being used more broadly than the trials is up to the regulators and physicians. We're a tiny NASDAQ-listed company, and so our goal is to do, a series of small, inexpensive, tightly scoped trials so that we can prove out the utility of the drug without wasting our investors' money on large trials that would normally be required for a new disease, but we believe in our case, because of the safety profile and the analogy with malaria, wouldn't be appropriate. But yes, to your point, like I have become, and I'm not a physician, as was noted from the discussion about my my grad training earlier in the talk, but I have come to believe, or personally have a more expansive view of presumptive treatment, because for two reasons. One, the diagnostics aren't very good. And then secondly, I think if your focus is acute disease, you don't think about the long-term consequences, or perhaps undervalue them. But when you're in your professional life, meet patients all the time, you have chronic diseases which don't have simple answers to. One approach is just to avoid getting that to start with, right? And presumptive treatment is one approach to that. And, you know, I've had not personal experience myself, but with a close family member who was bitten by a tick somewhere where there wasn't supposed to be any Lyme disease, got a rash, was very fortunate to have a dermatologist who knew exactly what it was and was on doxycycline straight away, and who knows what she avoided, because everything worked out right. But as you know from all the interviews you've had, that's not always the case. And so I take your point about having a more open-minded approach to how new drugs might end up eventually being used to treat all kinds of folks.
I'm sorry, Matt.
You know, I was just going to say, I think the reason I'm asking these questions is because when I knew I had Lyme disease and I was treated with just antibiotics for Lyme, nobody thought Babesia. Nobody even thought, what is the standard treatment for Babesia? But I was in such an immune compromised state. Would it have even helped me to have a Babesia diagnosis? Because likely it wouldn't have been able to effectively treat the Babesia. So I'm just thinking for our future, it's very hopeful to hear that with your research and your work, not only are we getting more awareness through a lot of just the work we're all doing as a community, but we're getting better drug development and testing as well. So if we're more aware and we have better treatments, when we do get diagnosed, we can hopefully get people better more quickly, or even more acutely if they get a tick bite, to be preventative. Because I do know a lot of what you did with malaria was, I think you had a prophylactic as well with malaria with some of your research, right? And what are your views on prophylaxis when it comes to babesiosis, when it, going into a tick endemic area, like going in a hike in a wooded area on Long Island, for example,
Yeah, so the first part of what you said, I agree with, and someone has to go first. So a lot of the lessons that we're learning as a company in drug development, so what is a case? What's the right trial to do? Is the FDA going to listen to what we have to say? What is the product profile you need? Like all of these basic drug development things, we have to prove that out because no one's ever done it before. So that's a challenge and an opportunity, and we've kind of waded in and are embracing that part of it, to the extent we can on a limited budget. So that's the first thing. As it relates to prophylaxis, there was a presentation at the recent tick-borne disease conference in Chicago by an investigator from Tufts University, Ed Veneer, and he showed in splenectomized mice, regular mice, and immunocompromised mice that if you inoculated those different groups of animals with Babesia and then a few days later treated them with a 3-day regimen of Tafenoquine, it completely eradicated their infection. And so the idea that Tafenoquine could potentially be used for prophylaxis, either post-exposure or pre-exposure, I think is, and then again, this is me speaking as a scientist and not a physician, I think it's a no-brainer from a scientific standpoint. But there haven't been any clinical trials to prove that out yet. And of course, those clinical trials would be very difficult to do.
So Dr. Dow, when you say no-brainer, and this is another one of the burning questions I had before we started the interview, I built out for you that, at least in our experience over the last 3 years for sure, that almost everyone who's chronically ill has the big three,
Yeah,
the Borrelia, Babesia, and Bartonella. And putting aside whether or not a clinician has the capacity to properly diagnose an acute illness, which is
yeah,
pretty rare. Your family member was very lucky to have a physician that had the clinical
yeah.
knowledge to even prescribe the doxycycline. But because ticks are in most cases, and certainly in many cases, harboring more than one microbe. And we're seeing that most people are becoming chronically ill because at least two bacteria and a parasite, and of course there are some viral infections that are being transmitted through ticks as well. Having a broad spectrum antibiotic is certainly wise in some cases for prophylactic treatment post tick bite. But it seems
Mm.
to me that it would also be wise to have some antiviral and some antiparasitic as a part of the standard treatment of care for someone who recently suffered a tick bite. So give me your reaction to that and how you think your clinical studies may put clinicians in a position where they can have a broader spectrum of tools to use, and perhaps the treatment protocol should be a cocktail rather than a single antibiotic.
So there's a company called Tarsus, and they are developing an oral product that kills ticks, and it can be administered after you've been bitten. And I think the idea is it kills the tick and prevents transmission of whatever they're carrying. So I don't know all the details of their program, but the point is conceptually, if there was a product that could kill the ticks right from the outset and prevent all the diseases, like it's a good conceptual thought from a product development perspective, and there are folks beginning to do some of that work.
So
Um,
um, meaning I take some form of a drug, and then when the tick bites me, the tick dies. Is that
yeah,
the
I mean that's how we stop dogs from getting tick-borne infections. We and heartworm infections and ticks and fleas is once a month, or on their collar they essentially taking prophylactic drugs which don't kill the diseases, they kill the ticks. And I'm just sharing that there's at least one company that's begun to think along those lines for human tick-borne diseases. Um, the the and if I could just maybe build out your thought some more, I think what you're proposing is that if someone was bitten by a tick and they went to the emergency room, there would be a standard protocol to treat those folks with a cocktail of drugs that was known or suspected to kill all the known pathogens that those ticks carry. I think that's what you're
it
suggesting
is. It is because
should be what the future is. All I can speak to is I can speak to two things: what we're doing, and then is what you're saying possible or feasible to do, and what some of the issues with it might be. So the animal data we've seen so far suggests that and this is all published, so I'm not sharing anything that's confidential. Same doses that prevent and cure Babesia infections in mice are pretty close to the doses that do the same thing in malaria infections in mice as well, right? And we've seen from the case studies in immunosuppressed patients that tafenoquine seems to add value and do things in combination with other drugs that the standard of care drugs don't do by themselves. So the overall hypothesis, which we're trying to prove, is that tafenoquine could work the same way in babesiosis as it does in malaria. And that includes the potential for some sort of preventative activity as well. But the issue with any sort of prevention drug is what's the regulatory pathway to proving it or getting the regulator to be comfortable with it. So from a safety perspective, we don't have an issue. From proving it in humans, we do, because in malaria, we could use a challenge model that there's no such thing in babesiosis. And the cost of doing a phase 3 study to prove post-exposure prophylaxis or pre-exposure prophylaxis for babesiosis would be way beyond the resources that a company that would be interested in doing it could probably raise. You're talking a minimum of $20 million. And at least for us, that's not feasible. And then I think that's true for Lyme. I think it's true for some of these weird, like Rocky Mountain spotted fever and Powassan virus. You're just never going to get enough cases to be able to definitively prove to the usual regulatory standard that the animal model predictions of what should work actually work in humans the way they're supposed to. So when we have that regulatory conversation with FDA, usually it's in the context of things like smallpox or Ebola or things where it was very difficult to do clinical trials, and then they have specific animal rule pathways where in theory you could use an animal model as a substitute for a clinical trial, but that is going to be a pretty involved conversation by a lot of people to get us to the point where you could get even one drug approved by that mechanism for a disease that's believed to be common enough that a trial is in theory feasible.
Well, I understand. I understand that it's going to be a large second step after folks like you who are doing the work in the Babesia arena to then take the next step to testing these drugs in combination when you have someone who is potentially infected with a combination of germs. So I understand that.
Well, I think that's doable in a treatment context, right? It's just hard to do in a prevention context. So as an example,
you mean treatment of somebody who's chronically ill as opposed to somebody who is not yet symptomatic but
yes,
tick
that's
bite?
exactly what I mean. Yeah.
Okay.
So for example, we've got a collaboration going with Tulane University where they're going to be looking at both Bartonella and Borrelia.
Who are you working with there? Dr. Embers?
Yeah, Dr. Embers. Yep.
She's awesome.
Yeah. And so our hope is, we've had people ask us, does tafenoquine work for Bartonella? And the short answer is, I don't know. So we funded a little bit of research that is being done in Monica's lab. And we're hopeful that if there's any interesting findings that come out of that maybe it offers an alternate route to a combination of tafenoquine with something else for either Borrelia or Bartonella. So, we've begun to think in that way, but I think the bigger problem is just the idea of doing clinical trials in chronic disease is, and this might be a good segue back into the trial we're going to start in October, but we only found we didn't have any problem finding clinical centers to do acute disease. So our hospital study, we've got 4 collaborating well-credentialed academic hospitals working on the program. And for our expanded access study, we've got two well-known research centers. We did a pretty good scrub. We were only able to find 1 clinical center that had the combination of the right clinical trial setting and experience, but interest and sympathy for patients with chronic disease who could do the chronic babesiosis study that we're planning at reasonable cost in a reasonable time frame. And I think that's just because the way the research community is oriented at the moment is acute disease where there's simple diagnostics, easily defined cases, and that's just the reality of the way clinical research is done at the moment. What we're trying to do is say, our patients are telling us chronic illness is an underserved problem. We see it every day and all the time. Let's try and include chronic disease patients as part of a development plan that includes the acute disease to keep all the folks who treat severe and life-threatening disease, on board as we move through the program and addressing that issue. But let's bring the chronic disease community along with us so that what we end up with is a product and approach that treats all patients. And that's kind of where we are in our thinking.
So I'd like to focus a little bit more on the chronic illness piece.
Yeah.
Do you believe that different people who are suffering from different chronic illnesses are going to have a different level of success with having their Babesia treated with the protocol? And I know I'm asking you to speculate, but the drug protocol that you're testing.
I think it's scientifically reasonable to think that there will be. Does Babesia cause chronic fatigue? Right? That's a question. Does Babesia make recovering from chronic fatigue more difficult? Is that same chronic fatigue exacerbated by a co-infection? So depending on what you think the answers to those questions are, and depending on whether the original illness was caused by COVID, whether it was caused by Lyme, whether it was Babesia by itself, yes, it's going to be a heterogeneous response. And perhaps if you've got a number of different chronic infections, you'll see less of an effect than if you only have Babesia, for example. The problem is you could spend months or years trying to slice and dice out the different subpopulations when we have to get back to the whole challenge in this disease space is I can't find the definition of chronic babesiosis in the literature. So why would I spend time trying to figure out the particular aspects of it that might be responsive to it's just very challenging. So the way we've decided to approach it is we've picked a common symptom that is not common to everyone with chronic tick-borne disease, but it's common to maybe 50% of folks with chronic disease. And then we're measuring the response and recovery from that particular symptom in a standardized way.
So, quick question, Dr. Dow. Just recently, a couple of months ago, the National Academies of Sciences and at the federal level came out with a publication, and our good friend Colonel Malachowski spearheaded that and was on the committee. And she renamed all these things because she felt that they were just so confusing. And she said, bottom line is they're chronic illness from infections. So the buzzword for the last 6 months or 8 months has been IACI, right? Infection associated chronic illness. What are your thoughts on that term? And have you heard it? And do you think that's something that will, take traction and pick up?
So I've come at that same thing in a different way. I think of all of these things as long diseases or long syndromes. Some case it's disease, some case it's a syndrome. You can debate about that. My thinking goes back to when that Nature paper was published in January of 2024, which described the pathophysiological process of chronic fatigue syndrome for the first time. And so essentially it's an acute illness, triggers an immune response, which is an immune overreaction. And that leads to a long-term dysregulation of the immune system, which has a CNS component. And then continuous antigen stimulation exacerbates it and makes it worse. And then, so the common theme is, yes, an insult, an infectious insult to start with, a dysregulated immune system, a set of chronic symptoms that overlaps regardless of what the initial infection was. There's a cognitive aspect to it, there's a fatigue aspect to it, there's a muscular aspect to it. And then particular variations of it depending on what is the persistent antigen that is continuing to stimulate and dysregulate the immune system, right? Is it leftover Lyme antigens? Is it a persistent Babesia infection? Is it a persistent Bartonella infection? We all can agree on what the end symptomology of that is, but the causes of it and how particular nuances of it affect different people, I think we're only starting to scratch the surface of that. And as a drug developer, we can only respond to the ecosystem as it is, right? So we, in developing the case definition for our chronic study, we originally focused on, we'll enroll anyone who's got a presumptive diagnosis of chronic babesiosis, because that reflects who the patients are that are being prescribed Arakoda off-label. As we consulted more and more with the FDA, with the ethics committee, with 2 panels of experts from ILADS and the Global Lyme Alliance, that case definition shifted to chronic fatigue that was disabling, some other symptoms of babesiosis, and then a lab diagnosis in the last 12 months that suggested exposure to the organism. But I don't know how representative of that, that tighter definition is to the original definition we had, which was a presumptive diagnosis from a physician. So that's the challenge. Is your question really gets to, how do we know that one drug is going to have benefit in everyone with chronic babesiosis? And the answer to that is it might not. On the other hand, we don't know enough about all of that to segment the population and target therapy. We're just not there yet in the science.
So, Dr. Dow, are there many people who are suffering from chronic babesiosis who are not also in conjunction suffering from what is loosely called a co-infection or another tick disease? How rare is that, or how often is it that patients are chronically ill just from Babesia?
So we only know anything about that in the context of acute disease. And what the data from hospital admissions tell us is that 40% of patients with babesiosis also have Lyme. And then when you do it the other way, what proportion of hospitalized Lyme patients have babesiosis? It's more like 10 or 20%.
But I'm asking the question the other way.
As it relates to chronic disease, I don't think there's a study in the literature that's prospectively gone out with the right diagnostics to answer the question you're posing, which is, when you talk about symptomatic people, how many of them have 1, 2, 3, or some other original cause for the chronic symptoms that they're experiencing. So certainly we hear from a lot of patients anecdotally that they've had chronic fatigue or chronic symptoms for 10 years. They've been through the Lyme protocols and they don't have Lyme anymore. And they're trying a new Babesia therapy because that's the next logical thing to target. Or sometimes it's with Bartonella. They'll have been through an antibiotic regimen for Bartonella. It hasn't resolved the symptoms. So then Babesia is the next thing that is being tried to address. But and so my perception is that there's a lot of polyinfections.
Yeah.
But I can't address that with data and facts because there is none in the literature because no one's ever done it.
Dr.
And
Dow.
that's part of the problem.
So I couldn't agree more that it's a poly problem, right? It's many things that are contributing to illness in many different people, and it's a combination of different things with different people. But I can't help but wonder, hydroxychloroquine is a drug that helps a lot of people in the chronic Lyme and chronic Babesia community. So I think its intended use is for autoimmune lupus, right? And yet it's used off-label by many doctors to treat Lyme patients as well. But I believe it's also an anti-parasitic as well, right? So what are your thoughts on that drug? Do you think that there's any kind of clues there between hydroxychloroquine helping people in the Lyme community, plus now your studies with Babesia and everything you're doing? What does that kind of give you just from a guess or a gut feeling standpoint, understanding that the science has to catch up and the studies have to catch up, but is there more there when you look at the big picture of other drugs too?
So if you think that the dysregulated immune system in Lyme disease is sort of like an autoimmune disease where the immune system is not performing the way it's supposed to, then using a drug that is known to have utility in treating arthritis and lupus, which are also autoimmune diseases, that makes perfect sense. And you could make an argument that there should be a clinical investigation into the utility of that product, specifically in post-treatment Lyme disease. That makes sense to me. I think based on the animal data, 4-aminoquinolines are, and when I say 4-aminoquinolines, I mean chloroquine, hydroxychloroquine, quinine, and mefloquine, are less effective than the standard of care. And when I say that, I mean azithromycin or atovaquone. And in the case of quinine specifically, it has a pretty nasty side effect profile that a lot of people find difficult to tolerate. And then with hydroxychloroquine, there's the or with hydroxychloroquine and chloroquine specifically, there's the risk of the retinal toxicity associated with long-term use. So it's not unreasonable to think that if it has an anti-parasitic mechanism and active Babesia prolongs people's chronic symptoms, that you might use something like chloroquine as an adjunct. But I think its real value is probably more as an immunosuppressive or immunomodulatory drug rather than its anti-parasitic effect. That's me speculating.
So, Dr. Dow, tell us more about the studies, because I know from reading up on you that there's a lot of stuff you have in the hopper, and you touched on it already. But tell us what's coming up in the next couple of weeks, what you've done already, and what you have in the hopper for the future, and what that means for everybody listening in the chronic Lyme and chronic Babesia community, and how these studies can ultimately lead to a viable approval potentially for a drug that can help them overcome chronic babesiosis. Because we hear it all the time on this podcast that Babesia is really difficult, and it's not talked about enough, but it's a very difficult, stubborn infection for people that are immune compromised.
Yeah, so we have a trial that will start in early November at Mount Sinai in New York, and it will enroll chronic babesiosis patients with a particular set of symptoms and medical history. And specifically, that's chronic fatigue that's disabling, some other symptom of Babesia, air hunger, anemia, and some lab evidence of exposure to Babesia in the last 12 months. And then we're going to test a 3-month regimen of Tafenoquine. So that'll be a loading dose over 4 days, and then 200 milligrams a week for 3 months. And we'll be measuring recovery from fatigue using a patient-reported outcome. So what that means is like just a survey specifically targeted and validated to assess quality of life and how fatigue is affecting daily living. And then the other thing we'll be doing is every month for the 3 months of therapy, and then for 3 months after, we'll be doing 3 molecular tests for babesiosis. One of them is the FDA-approved test that the blood bank uses to screen blood donors, and then 2 commercial PCRs, one of them from Galaxy Diagnostics, who you're probably familiar with,
Yep.
and then another one from Mayo Clinic. So those are the 3 diagnostics we're going to use. And that's important because it will accomplish 2 secondary objectives. One is to prove that someone who has a presumptive diagnosis of chronic babesiosis, we can actually prove they're infected with Babesia using a test that the FDA believes. And then the second value add is it will validate the commercially available diagnostics against that same test. So now moving forward, we'll have confidence that there's a drug that can potentially resolve fatigue in chronic babesiosis patients, potentially eradicate the infection, and it will give us a good preliminary sense of whether diagnostics that patients have access to right now can successfully be used to detect and monitor recovery from treatment in the future. So that's what we're trying to do with that study. It'll enroll about 100 patients, potentially up to 100 patients. It'll take about 12 months. And then it will be very interesting to see what the reaction from the mainstream medical community and the FDA is to that set of data. And then we'll take it from there in terms of what we do about follow-up studies or labeling.
So what I find really cool about that, and just correct me if I'm wrong with my assessment, this study is going to be using FDA-approved testing by two sound labs to prove that people in your study have an active Babesia infection. And then after using this Tafenoquine drug that you're trying to get verified for Babesia, you're going to retest and determine, okay, is the infection gone? And if we can prove that, then we can definitively say, and the FDA hopefully will approve, that this drug is a much more effective tool at treating Babesia than anything else on the market, and that we're recommending to treat people because they had a persistent infection despite treatment with other drugs that are our standard of drug. And now with Tafenoquine, we've been able to essentially get rid of it, and we have definitive proof. Is that a accurate assessment of what you just said?
That's the direction we're trying to head in and provide the data to support. And then I would just underscore that there's been so much scientific controversy in Lyme disease about whether the damn organisms are still there or not. And it's sort of strangled research, because from a patient perspective, maybe it doesn't matter if the infection is still there if you're experiencing chronic symptoms. And what we need is companies evaluating new therapeutics to treat the symptoms you actually have right now, right? What we're hoping to do is, by trying to prove that there's infection there, we'll know if it's there or not. And then we can proceed with, our drug's got an anti-parasitic mechanism. Awesome. Let's follow that pathway. If it's working through an immunomodulatory mechanism, that will take us in a different direction and provide the scientific clarity that we need to answer questions and provide new options for patients.
Yeah, and this is really promising. And in the last, I'd say, year, Rich, we've had a lot of promising interviews like this. So it's just giving me hope. And, I'm really, really just happy that we're having you, Dr. Dow, with all this research and pushing this to help the Babesia community. We have a ton of people helping on the Lyme and the Borrelia side. But I just want to touch on one thing, because you keep talking about chronic fatigue and how that is a common symptom of Babesia. But that's also another similarity with Lyme disease, right? Because the LymeDisease.org had their data-driven study, My Lyme Data, and the number one symptom, according to patient-reported symptoms for Lyme disease, was fatigue, right? So I just wonder how much of this fatigue is related to just the pool of polymicrobes, right? Meaning you have Lyme and Babesia, and if you're treating a persistent Babesia infection, maybe you're helping with that fatigue, and you're allowing the immune system to now do what it was supposed to do and address whatever's left, the Lyme and the other things. Because it's never just one thing, which makes me nervous. I guess that's where I'm going with this question, Dr. Dow, right? So why do you think that when you treat people with Tafenoquine, they have their fatigue lifted despite it being a polymicrobial problem? Does that make sense, the question?
The question makes perfect sense. And the short answer is we'll find out.
Yeah.
Right. This is what I know. If you treat an acute Babesia infection, fatigue resolves in most patients as your parasite load goes down, right? So is that true in chronic infection? I don't know. The patients tell us that, and again, this is anecdotal and doesn't prove anything from a regulatory perspective. It's just kind of a real world data points, is many people who've taken Arakoda to treat babesiosis feel better than when they started. And the question is, why is that? And it could be because it's an anti-parasitic drug that kills Babesia. It could be because it has an immunomodulatory effect that we don't know about. But if you don't run a trial and start somewhere, then all these questions will remain questions forever. So, you kind of got to put some lines in the sand and define things and collect some data, and then that gets the ball rolling, and then you go from there.
Yeah, I absolutely love that. And my final question for you, Dr. Dow, is the chronic Babesia and chronic Lyme community is not one you get into for fun, right? So you are clearly
Right.
passionate, you know what you know, and you are driven, and you have strong beliefs, and we're grateful for that. So, tell us, I guess, is there a personal history here for you that gives you this passion and this drive? Because I always sees Rich. I'm like, Rich, you don't have Lyme disease. Why do you care so much about this, right? So I can't help but wonder, seeing your passion, and everybody listening may not be able to see you. If they're watching on YouTube, they can see your face. If they're here listening on Apple Podcasts, they can't. But you are driven and compassionate, and you care, right? So what's behind that drive for you?
With malaria, it started. I spent a lot of time in Indonesia when I was a kid, and a lot of poverty and not everyone admires George Bush W, but I really think the best thing he did as president was PEPFAR and the aid program for HIV that saved millions of lives. And when I started out in malaria discovery and development, I had that same sort of desire to solve problems like that. That's why I originally started with malaria. And that same passion sustained years and years of slogging away getting a malaria drug approved. And then I guess the other thing is I'm an incredibly stubborn person, and when people say things can't be done, or they try and kill things based on no data, or they don't allow curiosity or questions to flourish because of hardened institutional positions. All of that stuff makes me incredibly mad. And so I guess the other driving thing is I really, and then there's another part of it. Like with malaria, 100 years of research, we've got to 3-day treatments. With babesiosis, we can't even, as a community of scientists and physicians, agree on what the case definition is. And the best treatment in a fully immunosuppressed person is months and months and months of poor quality drugs that are subject to drug resistance. And we just got to be able to do better than that. So Tafenoquine, I'm confident will get approved at some point for treating babesiosis, and that will be the first step. And then the step after that is, can we turn a regimen that will still take months of treatment into something that could be done in a week or less for everybody? And that needs to be where the gold standard is in the future, with what we're doing as a stepping stone to that, and perhaps across a broader range of vector-borne diseases as well, so Bartonella and Lyme and other things. So if I was going to rationalize an answer to your question, I think that collection of thoughts would cover it, Matt.
Yeah, and getting people like yourself focusing on Babesia and others focusing on Bartonella and Lyme, we could not be more hopeful and proud for the future of this community, because we know it's going to be drastically different in the short term. So this is a wonderful way to conclude this excellent interview, Dr. Dow. We cannot thank you enough for coming back for the long-form discussion here on our Full Tick Bootcamp podcast. And as you make progress, we'd love to have you back on down the road to share your amazing results, to continue to inspire and provide hope to everybody listening to this podcast.
Thanks, Matt. Thank you, Rich. I've enjoyed the discussion and looking forward to coming back and chatting with you soon.